Biological classification. We are going to learn here kingdoms like kingdom Monera, kingdom Protista, kingdom Fungi, Plantae, Animalia, viruses, viroids and lichens. From here we go for introduction. So it is given that Aristotle, he was a dawn of civilization. Next is Aristotle was earliest to attempt a more scientific basis for classification, means he has given a classification system. Aristotle was given a classification of system. He was also a dawn of civilization. He used a simple morphological characters to classify plant. He classified plants into trees, shrubs, herbs. He also divided animals into two groups: red blood, those who are having red blood, and those who did not have red blood. So he classified plants into trees, shrubs and herbs, and he also divide animals into two groups: the animals which are having red blood and the animals which are not having red blood. Aristotle is the dawn of civilization, and Aristotle has given the classification system. Okay, then next, two kingdom system was given by Linnaeus, and that two kingdom system includes kingdom Plantae and kingdom Animalia. The previously asked question, so please concentrate here. In Linnaeus time, a two kingdom system of classification with Plantae and Animalia kingdoms was developed that included all plants and animals respectively. So the question may ask like, who has given a two kingdom system classification? So the most correct answer is Linnaeus has given two kingdom system of classification that include kingdom Plantae and kingdom Animalia. The system, but what are the demerits of this system? This system did not distinguish between eukaryotes and prokaryotes, unicellular, multicellular, photosynthetic and non-photosynthetic organisms. Then next, table characteristics of five kingdom system. R.H. Whittaker in 1969 proposed a five kingdom classification. This is the most important question for exam point of view and asked many times previously. Who has given a five kingdom system classification? The answer is R.H. Whittaker in 1969 proposed a five kingdom classification. It includes Monera, Protista, Fungi, Plantae and Animalia. The main criteria for this classification used by him include cell structure, body organization, mode of nutrition, reproduction and phylogenetic relationships. It may also ask what are the main criteria for five kingdom system. So you must know the main criteria are: first, cell structure; second, body organization; third, mode of nutrition; fourth, reproduction; fifth, phylogenetic relationships. So let's learn table 2.1 characteristics of five kingdom system, and this five kingdom system was given by R.H. Whittaker in 1969. The characters, see in this table 2.1 characters, in that there is cell type and five kingdoms are written like Monera, Protista, Fungi, Plantae and Animalia. So cell type is in Monera it is prokaryotic, in Protista it is eukaryotic, in Fungi it is eukaryotic, Plantae eukaryotic and Animalia eukaryotic. So the cell type of Monera is prokaryotic, Protista is eukaryotic, Fungi eukaryotic, Plantae eukaryotic and Animalia eukaryotic. The cell wall of Monera is non-cellulosic, Protista present in some, Fungi present with chitin. The cell wall of fungi have chitin. So question is asked earlier, the cell wall of fungi contains. So cell wall of fungi is made up of chitin. Then next, cell wall of plant is present with cellulose and cell wall of animalia is absent. The nuclear membrane in Monera, nuclear membrane is absent. In Protista, fungi, plantae and animalia, nuclear membrane is present. Next is body organization. Body organization in Monera is cellular. Protista also cellular. In fungi, body organization is multicellular with loose tissue. In plantae, tissue organ system. In animalia, tissue, organ and organ system. Mode of nutrition. How the Monera take nutrition? It is autotrophic, means can prepare their food by self. It is called autotrophic. Protista is also autotrophic. Fungi is heterotrophic. Fungi depend on others for food, so it is heterotrophic. Then plantae, it is autotrophic. They are photosynthetic. They use sunlight for energy, so it is photosynthetic. Animalia, animals are heterotrophic. So in this way we completed this table 2.1. Go through it. It's very important for question point of view. It may ask question from anywhere. They can ask about fungi, cell wall of fungi. They can ask about nutrition of fungi. They can ask about body organization. So go through it, write it once and learn it. Earlier classification system include bacteria, blue-green algae, fungi, mosses, ferns, gymnosperm, angiosperm under plant. Earlier they had given under plants like they included in plants bacteria, blue-green algae, fungi, mosses, ferns, gymnosperm, angiosperm, etc. Now it has separated. So the character that unified this whole kingdom was that all organisms included had cell wall in their cells, hence placed together with widely differing other characteristics. Why these were kept in plants? Because all were having well developed cell wall, so they were placed in plants. It brought together prokaryotic bacteria and blue green algae with other group which were eukaryotic. They brought together prokaryotic bacteria and blue green algae. Blue green algae is also called as cyanobacteria. They were kept together which were eukaryotic, means they want to separate the group prokaryotic and eukaryotic, but bacteria was kept with prokaryotic and eukaryotic ones. It also grouped together unicellular organism and multicellular organism once. Means they kept unicellular as well as multicellular in plants only. For example, Chlamydomonas and Spirogyra were placed together under algae. So these are the two different organisms. One is unicellular and one is multicellular. So they were kept under algae. So these are the demerits. The classification did not differentiate between heterotrophic group fungi and autotrophic green plant, though they were showed a characteristic difference in their cell wall composition. Fungi had chitin in their wall, while green plant had cellulosic cell wall. Means they are saying they have differences in their cell wall. Fungi is made up of chitin and plants are made up of cellulose. So then also they were kept together in same group. This is also a demerit. Which such characteristics were considered, fungi were placed in separate kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and unicellular eukaryotic organisms were placed in kingdom Protista. So after this, all prokaryotic organisms were placed in kingdom Monera. Means kingdom Monera contains all prokaryotic organisms, while kingdom Protista contains all eukaryotic unicellular organisms. Kingdom Protista has brought together Chlamydomonas, chlorella with Paramecium and amoeba. Chlamydomonas and chlorella were earlier placed in algae with plants and both have cell walls. And Paramecium and amoeba which were earlier placed in animal kingdom which lack cell wall. It has put together organism which is earlier classification were placed in different kingdom. This happened because of criteria for classification changed. Over time, an attempt has made to evolve classification system which reflects not only morphological, physiological and reproductive similarities but also phylogenetic, that is based on evolutionary relationship. So let's learn kingdom Monera. I told you earlier all prokaryotic organisms were grouped together under kingdom Monera. So before learning this, you must know kingdom Monera includes all prokaryotic organisms, while kingdom Protista include unicellular eukaryotic organisms. So let's learn kingdom Monera. Kingdom Monera. Bacteria are the sole members of kingdom Monera. Most important line for question point of view. They may ask sole members of kingdom Monera. So you must remember that bacteria are the sole member of kingdom Monera. In Monera there are bacteria and they are the sole members. They are most abundant microorganisms. We are knowing about the bacteria. Bacteria are present everywhere. They are omnipresent everywhere. So they are the most abundant microorganism. Bacteria occur almost everywhere. Hundreds of bacteria are present in handful of soil. Means many bacteria, abundant bacteria are present in soil. They also live in extreme habitat such as bacteria can live in hot spring, desert, snow and deep oceans which very few other life forms can survive. Means bacteria can survive anywhere. They can survive in warm places also, in cold places also, in oceans also. Anywhere bacteria can survive. So it is written that you must know this scientific line of NCERT that such as they can live in hot spring, desert, snow, deep oceans which vary from life forms can survive. Many of them live in or on organism as a parasite. These bacteria can be present in human beings inside the intestine. So they are called as parasites. Means the bacteria which depend on live organisms are called parasites. Bacteria are grouped under four categories on their shape. Bacteria have four shapes, that is coccus. Coccus means spherical. The bacteria shape is spherical. See the diagram. The cocci is shown here. So coccus means spherical. It is rounded bacteria. Coccus means cocci. Plural it is cocci. It is rounded bacteria. Second is rod shape. That is called bacillus. Bacilli is rod shaped bacteria. And third one is comma shaped. Comma shaped bacteria is called Vibrilum. Means Vibrio. Vibrio is called comma shaped bacteria. See the diagram number 4. It has given comma shaped bacteria, that is called Vibrio. And fourth is spiral, that is called Spirulum, Spiralia. Okay, you can refer this diagram for various shapes of bacteria. You have to learn spherical, coccus, rod shape, bacillus, comma shape, Vibrium and spiral, Spirulum. They may ask question like what are the different shapes of bacteria. They may have given what are the scientific names of various shapes of bacteria like spherical, coccus, comma shape, vibrio. So you must remember all these four types. Though bacteria structure is very simple, they are very complex in behavior. Bacteria structure is very simple. Bacteria is very simple in their structure, but it's very complex for human being. Compared to many other organisms, bacteria as a group show most extensive metabolic diversity. Some of the bacteria are autotrophic. They synthesize their own food from inorganic substrates. So what is meant by autotrophic? Autotrophic means they can prepare their food by own, means synthesize their own food from inorganic substrates. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, that is, they depend on another organism or dead organic matter for food. Most of the bacteria are heterotrophs. Heterotroph means what? They depend on others for food. So they may be depend on live organisms or on dead organic matter for food. So this is Monera. Let's revise. You have to keep in mind about Monera is Monera are the sole bacteria are the sole member of kingdom Monera. Means you have to keep in mind that whenever their word comes kingdom Monera, you must think about bacteria. Once you think about bacteria, then you will come to know what is there in bacteria. Like they are the sole members of kingdom Monera. They present everywhere. They can be found in hot spring, deserts, snow, etc. And their four shapes like coccus, bacillus, vibrilum and spirillum. Okay, then next bacteria is archaebacteria. These bacteria are special since they live in some of most harsh habitat. These bacteria live in most harsh habitat like salty areas called halophiles, hot spring, thermoacidophilus, marshy areas, methanogens. So these bacteria live in harsh habitats. Extreme salty area, the bacteria which live in extreme salty areas that are called halophiles. The question asked, bacteria which live in salty areas are called, they may give four options: halophiles, acidophilus, methanogens, all of the above. So you must know the most correct answer, that is halophiles. Then next is they live in hot spring that are called thermoacidophilus. Third is marshy areas. Marshy areas means methanogens. Archaebacteria differ from other bacteria in having different cell wall, and this feature is responsible for survival of extreme conditions. Means archaebacteria have well developed cell wall. Because of this different cell wall, they can survive in extreme conditions. So, methanogens are present in gut of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane biogas from dung of these animals. So, methanogens which we have learned earlier, marshy areas have methanogens bacteria. Methanogens are present in gut, in throat of these animals such as cows and buffaloes. There they are responsible for the production of methane. They produce methane biogas from dung of these animals. Very important, which I have underlined all these by red and green. It is very important for exam point of view. They may ask question from anywhere. So methanogen produce which gas? So answer must be methane gas. methane gas, from which material? So you must know it is prepared from dung of animals like cows and buffaloes. They are present in ruminant. They are present in gut of the several ruminant animals like cows and buffaloes. You must know this. You must remember about extremely salty areas called as halophiles, hot spring called as thermoacidophilus, marshy areas like methanogens, etc. So these were about archaebacteria. So let's learn eubacteria. Most important line of this eubacteria are, it is a true bacteria. Eubacteria means what? It's true bacteria. There are thousands of different eubacteria. They are characterized by the presence of rigid cell wall and motile flagellum. This eubacteria have very different cell wall. It have rigid cell wall and has flagella. It contains flagella which is motile. The cyanobacteria also called as blue-green algae. Keep in mind, whenever this cyanobacteria come, you must remember about blue-green algae. It have chlorophyll A similar to green plants and are photosynthetic autotrophs. Cyanobacteria, these are also bacteria which are blue-green algae. Earlier we have learned the bacteria are heterotrophic, means they depend for food on others. So cyanobacteria is one example which is photosynthetic autotroph. Photosynthetic means what? They take energy from sunlight for the production of food. So it is called photosynthetic autotroph and it has chlorophyll A like green plants. And cyanobacteria it is also called as blue-green algae. The next is the cyanobacteria are unicellular, colonial or filamentous, freshwater or marine or terrestrial algae. Cyanobacteria are unicellular, made up of single cell, colonial, filamentous algae which is present in fresh water as well as marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. The colonies of this cyanobacteria is surrounded by gelatinous sheath. They often form bloom in polluted water bodies. Sometimes in polluted water it forms blooms. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts. Example, nostoc and anabaena. Means some of this cyanobacteria has ability to fix atmospheric nitrogen. Means they take atmospheric nitrogen in specialized cells. They have some specialized cells called heterocysts. So let's see in this figure 2.2. Here it is given that heterocyst C and their body has mucilaginous sheath, which we had discussed earlier, that the colony is generally surrounded by gelatinous sheath. This gelatinous sheath is also called as mucilaginous sheath. So see this mucin is surrounded all over the body. So you have to keep in mind. They may ask a labeling of this diagram. They may give simply diagram and they will ask to label this, and you have to mark out the correct option from this. So listen and see carefully. Heterocyst is given and mucilaginous sheath is also shown here. So what is heterocyst? Heterocyst means what? The organism can fix atmospheric nitrogen into the specialized cells. These cells are called heterocysts and the examples are given like filamentous blue-green algae, nostoc and anabaena. These are the two examples. Examples of heterocyst is nostoc and anabaena. And what is meant by heterocyst? These are having ability to fix atmospheric nitrogen in the cells. They are called heterocysts. Examples are nostoc and anabaena. Most important. Then, chemosynthetic autotroph bacteria oxidize various inorganic substances such as nitrate, nitriles, ammonia and use the released energy for their ATP production. Here, chemosynthetic autotrophic bacteria. They oxidize inorganic substances such as the inorganic substances which are present here. They oxidize them such as nitrates, nitriles and ammonia and use their energy for ATP production, and they play great role in cycling nutrients like nitrogen, phosphorus, iron and sulfur. They may ask question, chemosynthetic autotrophic bacteria play great role in recycling nutrients like, you have to learn these four: nitrogen, phosphorus, iron and sulfur. Next question may ask like, what are the various inorganic substances used to oxidize chemosynthetic autotrophic, used to oxidize, which were oxidized by chemosynthetic autotrophic bacteria? So you must know this inorganic substances like nitrates, nitrites and ammonia. Okay, next is heterotrophic bacteria. Heterotrophic bacteria are most abundant in nature. Most abundant means almost 90% bacteria are heterotrophic. The majority are important decomposer. Bacteria used to decompose. Decompose means what? They used to, कोई भी चीज को सड़ाने के लिए जो बैक्टीरिया होते हैं, they are called decomposer. Okay, they make decay. They may present in dead body. They may present in moist places, everywhere. Many of them have significant impact on human affairs. They are helpful in making curd from milk. Yes, bacteria are useful for making curd from milk. Production of antibiotics. Fixing nitrogen in legume. So you have to learn these examples. Heterotrophic bacteria are useful for making curd from milk. One first example. Second is for the production of antibiotics. Third is for fixation of nitrogen in leguminous roots. Some of the pathogens causing damage to human beings, crop, farm animals and pets. Some of these also cause damage to crops, farm animals and pests. Chlorella, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. Some bacteria may cause disease like typhoid, tetanus, chlorella, citrus canker, etc. You have to keep in mind these four diseases which affect human being by various bacteria, that is chlorella, typhoid, tetanus and citrus canker. Then bacteria reproduce mainly by fission. So let's see the example. Figure number 2.3. So bacteria is having cell wall, cell membrane, and inside the cell membrane there is genetic material given, that is DNA. So see the division of this bacteria, a dividing bacteria. Okay, bacteria reproduce mainly by fission, sometime under unfavorable condition. They produce spore. They can reproduce by fission and sometime in unfavorable condition by production of spores. They also reproduce by sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to another. They may, means you have to keep in mind that bacteria can reproduce by fission sometimes by spores, and sexual reproduction also by adopting primitive type of DNA transfer from one bacterium to another. Now, mycoplasma. Mycoplasma are the organism that completely lack cell wall. So from this chapter biological classification, you have to keep in mind this is the most important question they can ask in exam. Mycoplasma are the organism that completely lack cell wall. They may ask like which is the organism which completely lack cell wall, and you have to click this mycoplasma. They are smallest living cell known can survive without oxygen. This is also most important thing. You have to learn by heart this. This is the smallest living cell. Living cell can live without oxygen. Many mycoplasma are pathogenic in animals and plants. First thing about mycoplasma, they completely lack cell wall. Second is can survive without oxygen. So we had learned kingdom Monera. In this kingdom Monera,